Bismillahirrahmanirrahim. Dear students, welcome back. In the previous lecture, we were discussing the different characteristics of enzymes. In the continuity of our previous lecture, we were discussing the cofactor. First of all, what is cofactor? The non-protein part of enzyme is called as cofactor. So the question arises in the mind of every student that whether every enzyme needs another partner, another actor to perform its function. So my answer is yes. There are some enzymes which need a partner, an actor, which facilitates its activity, and that is called as a cofactor. And an enzyme without a cofactor is called as apoenzyme. A very popular example is the RNA polymerase 2, which have 4 subunits. One is called as sigma factor. Without the sigma factor, the coenzyme is called as apoenzyme, and if we remove the cofactor, this is apoenzyme, and if we add the cofactor, then this is called as holoenzyme. So dear students, cofactors are of 2 types, organic and inorganic. I will not go in details about what is organic, but basically those compounds which consist of carbon, hydrogen and oxygen in specific proportions, we call these chemicals as organic. And inorganic are those chemical substances in which usually there is no specificity of carbon, hydrogen and oxygen. The example are magnesium and manganese. I mentioned here calcium is also a cofactor. Magnesium, I have written here magnesium. Magnesium is the cofactor of about 70 plus enzymes, in which one of them is hexokinase enzyme hexokinase. So my dear students, what is hexokinase? Inshallah, when we will explain chapter 7, in chapter 7 in respiration, instead of the respiration, the first step is the glycolysis, and in the glycolysis, the first reaction is the conversion of glucose into glucose 6-phosphate. And my dear students, for this catalysis, we need hexokinase enzyme. Hexokinase is the enzyme which catalyzes that particular reaction. The second one is organic. The organic cofactors are divided into 2 types, prostatic group and coenzyme. Dear students, if the organic group is tightly attached with enzyme, then this is called as prostatic group. But does it mean by tightly attached? Dear students, if prostatic group is covalently bounded with the enzyme and with the removal of prostatic group, the enzyme denature. This is called tight bonding, and this is the meaning of tightly attached. So if we remove the prostatic group from the enzyme will definitely be denatured. The example is heme. What does it mean by heme? All of you know RBC. On RBC, red blood cell, there is a compound called hemoglobin. In the hemoglobin, we have 4 heme groups, which are iron center having 4 nitrogens on its sides. This is called a heme group. The heme is a good example of the prostatic group. The next is the coenzyme. Coenzymes are those cofactors which are loosely attached with the enzyme. Examples are NAD, FAD, folic acid. What is NAD? Nicotinamide adenine dinucleotide, and FAD, flavine adenine dinucleotide. Dear students, NAD and FAD are performing their function as they accept electrons and they are popular electrons carriers in the cellular respiration process, in the glycolysis, in the Krebs cycle, in the electron transport chain. We will study the role of NAD and FAD. Usually they carry the electrons. Dear students, the last characteristic of enzyme is the metabolic pathway, that how enzymes act in a metabolic pathway. So dear students, one concept you have to keep in your mind, that is, if I have a substrate A and substrate A is converted into product and this is B, E1 is the enzyme which catalyzes the conversion of substrate A into B. So in the next step for E2 enzyme, this product is a substrate. It will convert it into a product called C, but the C is the substrate for E3 and it will convert it into product D. So in a metabolic reaction, if you have a chain of reaction, one enzyme is converting a substrate into product, the next enzyme, that product will be a substrate and it will convert it into another form, and so on. These metabolic pathways are very common in the chain metabolic reactions like cellular respiration. In glycolysis, we have 10 steps of different chemical reactions that take place in the glycolysis process. So this phenomenon is applied. So these were all the characteristics of enzyme. First characteristic was it is protein in nature. Second was the speed of the enzymatic activity, that without enzyme and with the enzyme, how what should be the probable speed of the chemical reaction. The active sites we discussed, cofactors we discussed, inhibitors and activators we discussed, and finally we came to the metabolic pathways. It was all about the characteristics of enzymes. The next topic are the uses of enzymes. Dear students, enzymes are not only very important for our body, metabolic activities, metabolic reactions, but also they are extensively used in different industries. Like in food industry, they are used to make the conversion of starch into sugar. Usually amylase enzymes are used in this particular industry which converts starch into simple sugar. White bread, buns are used. The beverage industry, beverages industry, alcoholic drinks, alcoholic beverages are made by the process of fermentation. Fermentation is actually anaerobic respiration which occurs in the absence of oxygen, and this function is performed by the yeast or certain bacteria can do this, and simply the glucose is converted into ethyl alcohol plus carbon dioxide. This is called fermentation process, and in the alcoholic beverages industry it is widely used. In the paper industry, how paper is made? Usually paper is made from the starch, and for this purpose we need to look down the viscosity of the starch to make a paper. What is viscosity? Viscosity is actually the rate of the flowing of a fluid. So it is if a substance is more viscous, it means it should be hard to flow. It's a resistance to flow actually. So it starts. We have to dilute the starch. We have to load out its viscosity, and then we can make paper. The biological detergent. It's also used as a detergent because if you have protein stains, so you can use protease. If you have carbohydrate stains, in dish, in the detergent which is used for the dish washing purposes, the detergents are used having amylase and mainly, and lipase is also to remove the lipid stains. So different stains made by different organic substances use particular enzyme as a detergent. So we can use these detergents, these enzymes as a detergent to remove the particular stains. Dear students, the next topic is the factors affecting the rate of enzyme action. Dear students, enzymes are very sensitive to the environment in which they are working. They are sensitive to pH, temperature, they are sensitive to pH. Many factors are affecting the rate of enzymatic activity. First of all, let's study the effect of temperature on the enzymatic activity. Increase in temperature speed up the enzyme action. I have written a very small word here, in limits. What does it mean by limits? If we increase temperature in limits, it will increase the enzymatic activity, and there will be a temperature on which enzyme will work very best. That temperature is called as optimum temperature. After the optimum temperature, if we are enhancing the temperature, then the rate of the enzymatic activity will decrease. And here I wish to mention a term called denaturation. If I increase the temperature from the normal limits, from the optimum limit, then the denaturation will start. What does it mean by denaturation? Denaturation means the disturbance of the three-dimensional structure of the molecule of the enzyme is called as denaturation. Here a graph is given. These are the human enzyme example. If I am increasing temperature, the rate of enzymatic activity is increased, and on 37 degrees centigrade, the enzymatic activities are in its peak. But if I am enhancing the temperature further on, the rate of the enzymatic activity is decreasing and reaching to the 60 degrees centigrade. The enzymatic activity ceased completely because all of the molecules of the enzymes are denatured. Next is the pH. All of you know very well that this is the potential of hydrogen, and low pH means acidity and high pH means a basic property. Just like the temperature sensitivity, they are also sensitive to pH. But the pH effect is not uniform at all, because some enzymes work best at low pH and some enzymes work best at high pH. For example, pepsin, which is a popular enzyme that is present in the stomach for the digestion of protein, works best at pH 1.7. Some books are writing 2 also. And trypsin, which is an enzyme of the pancreas and working in the small intestine, work best at 7.7 pH. So here the optimum pH is that pH on which an enzyme work best or enzymatic activity is the best. That is called optimum pH. If we increase or decrease the pH from the optimum limits, then again the denaturation of the three-dimensional structure of the enzymatic molecule will happen and it will decrease the enzymatic activity. Dear students, the next factor which is affecting the rate of enzymatic action is the substrate concentration. In my previous lecture, I explained in detail that what is substrate actually. What is substrate? Substrate is any substance upon which enzyme is acting. That particular chemical substance is called substrate. If enzyme molecules are available, the substrate in a medium, the substrate concentration increase enzymatic activity. But when all active sites are occupied by the substrate, addition of further substrate, there will be no change in enzymatic activity. In this graph, if you see that I am adding the substrate, so enzymatic activity is increasing. But when the saturation level comes and all active sites are occupied, then there is no net change in the enzymatic activity. Dear students, these are the three factors which are included in your syllabus, which are affecting the rate of enzymatic activity. First was temperature, then pH, and now substrate. So these are all factors which affecting the rate of enzymatic activity. Dear students, our next topic is the mechanism of enzyme action. How enzyme is acting on the substrate. And so first of all, enzyme plus substrate, enzyme attached to a substrate, they are making a temporary complex called enzyme substrate complex, IES complex. And in the next step, we get the product and enzyme comes out, as we say. We explained to you earlier that actually enzyme is acting here as a catalyst. So the catalyst is just to speed up the chemical reaction and itself unchanged, unused. You can see here enzymes came out from the reaction. Dear students, there are two models which explain the enzyme action mechanism of enzymatic activity. One is the very old model, lock and key model. Emil Fischer proposed this model in 1894. And according to this model, the active site is rigid and non-elastic, just like the key and lock. If you put the right key in the lock gets open. But if you put the wrong key, then the lock will not get open. In the same way, the shape of the substrate must be according to the shape of the active site, then ES complex can be formed and the reaction can proceed. But if the shape of the substrate is different from the shape of the active site of the enzyme, then ES complex will not form. And the second model is the inducement model. In 1958, is American biologist, and this is also called Koshland hypothesis. When I was a student, it was a hypothesis, and we always called it as a Koshland hypothesis. But now it's a model, and Daniel Koshland was the man who proposed this model. According to this model, the enzyme active site will be elastic and it can change itself according to the shape of the substrate. Look at this enzyme, its active site is different from the substrate, but enzyme active site change itself according to the substrate and ES complex is formed. Dear students, so So this is the main difference. Here the active site is rigid, non-changeable, and it will fit only with those substrates who have the same shape according to the active site. And here in induced models tells us that the active site can be changed according to the shape of the substrate. So the final thing is, which one is the best? I will say both are the best. On some enzyme, lock and key model is best, like urokinase enzyme. On urokinase enzyme, this model is applied in very good manner. And in some enzymes, induced rate model, so both of the models are applicable and applied. Dear students, the last topic of this chapter is enzyme specificity. Dear students, enzyme specificity, in my previous lecture, I told you that enzymes are really specific in the characteristics of enzyme. I explained it. But in protease enzyme, like I told you, that protease enzymes will work only on protein. Amylase enzymes will work only on starch. Like DNA polymerase enzymes will work only for the DNA replication. The RNA polymerase enzymes will work only for the synthesis of messenger RNA. So dear students, every enzyme is specific for their specific substrate. This is called enzyme specificity. There are 2000 plus enzymes. Some books are writing 3000 plus enzymes are present in a system. Dear students, there are two types of enzymes basically. Extracellular and intracellular. Extracellular are pepsines. Pepsine is a good example. Trypsin is a good example. Lipase is a good example, which usually secrete inside in a cavity. Like pepsine is secreted in the stomach, and trypsin, amylase, and lipase are secreted in the small intestine, duodenum part for the digestion purpose. Along with that, certain enzymes are intracellular, like enzymes hexokinase, like ATPase enzymes. These are enzymes which are present inside the cell and performing their cell functions inside the cell. Thank you so much, and hope that you have learned this chapter in a very good way. We are waiting for your feedback on online question answer session. Thank you so much.